Today, in part 4 of Unit 5, we examine non-Mendelian genetics. That is, traits that do not necessarily follow Mendel's rules and laws of heredity. It turns out there are a great many genes that do not behave the way Mendel's pea plant genes behaved. And you might wonder, if Gregor Mendel was so wrong about so many different types of genes, how can we still consider him to be the father of genetics? And the reason is simple. Absolutely nothing that we currently know about genetics could have been known, could have been figured out without the foundation that was created by Mendel's work. Without Gregor Mendel's work, we simply would not have known where to start in our understanding of how heredity works. So we still owe Gregor Mendel a tremendous debt of gratitude. And there are certainly many genes that do work exactly the way Mendel described in his 1865 research paper. However, it is important to understand some of the exceptions to Mendel's rules, and that's what we will delve into today. So every trait that we've discussed previously, all of the pea plant traits and human PTC tasting and other things that have come up in previous lessons, have fallen into the category called complete dominance. And this follows Mendel's rule of dominance, which says that if an individual inherits two different alleles for a gene, like big P for purple and little p for white flowers, that only one of the two alleles will be expressed. In this case, the dominant purple allele is expressed, and this plant, this heterozygous big P little p plant, has purple flowers, and they're just as purple as the big P plant. So those the phenotype for big P is indistinguishable from the phenotype of a heterozygous purple plant. There are exceptions to this rule. There are genes that follow the incomplete dominance pattern. And that means that there are two different alleles, but neither one is dominant and neither one is recessive. So this is new. We haven't talked about this before, but the thing about incomplete dominance is the hybrids or the heterozygous individuals, the F1 hybrids, have a phenotype that's intermediate. It's in between the phenotypes of the two parents. And a wonderful example of this is in a species of decorative flowers that people often plant in their flower beds in their yards, called a snapdragon plant. I'll show you a great example with snapdragons of how incomplete dominance works. There's another category, another degree of dominance, you might call it, called codominance. And codominance occurs when there are two different alleles that can be inherited. And if an individual is heterozygous, and if that individual inherits both of those dominant alleles, because they're both dominant, they're both expressed equally. Neither one can cancel out the other. They both affect the phenotype. A great example of that is the way humans inherit blood type, like type A blood or type AB blood and so forth. And I'm going to use that as my example of codominance. I just realized I misspelled the word dominance here, so let me pause and correct that because it's driving me crazy. Okay, so that's much better. Notice I have corrected my spelling mistake. And I think both of these concepts, incomplete dominance and codominance, are going to make a lot more sense to you when we look at some examples. So let's do that. So incomplete dominance, as I said, is where heterozygous individuals show an intermediate, sort of in-between phenotype. And this, when you look at this, it's going to seem like it's blended inheritance. It's going to seem like it violates Mendel's concept of particulate inheritance. But in fact, it does not. This is not a violation of the particulate inheritance hypothesis. And I can prove that to you in a moment. But we're going to use the example of snapdragons, which again are decorative flowering plants. So here's the cross. In the parental generation, we're going to take a homozygous red-flowered snapdragon. Look how lovely this plant is. And we're going to cross-pollinate that with a white-flowered snapdragon. Now, if Mendel had done such an experiment, if he had experimented with snapdragons rather than garden pea plants, he would have expected the F1 generation to have red flowers, right? Because he probably would have assumed that red is dominant and that white is recessive, and so therefore the F1 offspring should all have red flowers. That would be the pattern that he saw in pea plants. That is not what happens with snapdragons. In the F1 generation, all of the offspring are heterozygous or hybrid, we might call them, pink flowered plants. And so you see, this looks like blended inheritance. It looks like the allele for red has blended with the allele for white to produce a pink allele. And this would have upset Mendel greatly, because he spent a great deal of energy and collected a great deal of data proving that blended inheritance is wrong. But this is not blended inheritance, because there is no such thing as a pink allele. There are red alleles and there are white alleles. And even though this looks like blended inheritance, in fact, it's not. I'll show you how I know that. If you cross pink with pink, then you get an interesting result. In the F2 generation, you get some flowers, or some plants that have red flowers, some that have pink flowers, just like the parent here, and some that have white flowers. If blended inheritance had occurred, we should see nothing but pink in the F2 generation. But clearly, the allele for red and the allele for white remain distinct and separate, and they are passed down not in a blended state, but as separate particles. So this, in fact, is not a violation of particulate inheritance, but it still would have baffled Mendel, because clearly the red is not dominant and the white is not recessive. So what the heck is going on here? Well, it turns out that there are two separate alleles, R for red, as you would expect, and then we use this R prime. It's an R with a little apostrophe. R prime is the allele that codes for white. Notice I'm not using lowercase r, because what would that imply? A lowercase letter would imply that, it's recessive. And we know that's not the case. Some textbooks will use a capital W for white, which probably makes more sense, but this is kind of the way you see it most often in college textbooks, so we'll stick with that. So a red flowered plant is homozygous, RR. A white flowered plant is homozygous R', and when you cross-pollinate these two, this parent has to pass down an R allele, this parent has to pass down R', and so what in fact you have, there's no such thing as a pink pigment. This plant produces the same red pigment as this plant, but it produces half as much. So even though this plant is only producing red pigment, there's no such thing as a white pigment, the R' allele codes for no pigment production. So you've got one allele coding for red, another allele coding for lack of pigment, and the end result is the plant is going to produce red pigment, but only half as much. And when you spread out that red color over a large area, it looks pink. So if we cross pink with pink, that's RR prime crossed with RR prime, and you see the result. 1/4 of the offspring will be red, RR. Half of the offspring will be pink, that's heterozygous, and 1/4 of the offspring will have white flowers, R prime, R prime. Now let's find out if you truly understand how incomplete dominance works. Let's look at a problem. Show the cross between a red flowered snapdragon and a pink flowered snapdragon. And we're going to use a Punnett square for this. And after we complete our Punnett square, we're going to answer the question, what phenotypes, in other words, what color flowers, are expected in the offspring? So here's my red flowered snapdragon, RR, crossed with a pink snapdragon, which is R prime. Remember, it has a red allele and a white allele. That's what makes it have pink flowers. And so if we cross this red plant with the pink plant, we would expect half of our offspring to have red flowers, and we would expect half of the offspring to have pink flowers. And so this is the way we would write our expected genotype and phenotype results. So again, I'm just using fractions to reflect what I see in my Punnett square. So half of the offspring are expected to be homozygous R, half of the offspring are expected to be heterozygous, and then phenotype refers to, what are they going to look like? Well, I'm expecting about half of them to have red flowers and half to have pink. Okay, let's look at another incomplete dominance problem. This one also deals with snapdragons. 2 snapdragons are crossed. Now, right off the bat, notice I'm being purposely vague here. I'm not telling you what color the parents are. You have to figure that out. 2 snapdragons are crossed. Maybe it's a red crossed with a pink, or maybe it's a pink crossed with a white, or a red crossed with a white. Who knows? We got to figure it out. But what it tells us is the offspring. There are 64 offspring. Plants make lots of seeds, so that's lots of baby plants. Of the 64 offspring, 31 of them grow up to have white flowers, and 33 of them grow up to have pink flowers. Interesting. So what I'd like you to do is see if you can show me the cross, show the Punnett square. You can work this out on a piece of scratch paper or right there in your notes. See if you can show me the Punnett square that produced these offspring. This would be a good time to pause the video, and then after you work your Punnett square, you can check your answers. Okay, so this one we have to kind of think backwards. It doesn't tell us what color flowers the parents have, but it does tell us something about the offspring. Approximately half of the offspring, I know 31 is not exactly half of 64, but it's pretty close. Approximately half of the offspring have white flowers, and approximately half have pink flowers, which means that half of the offspring should be R prime, R prime. Those are the ones that have white flowers. And half of the offspring should be heterozygous. Those are the ones that have pink flowers. So when I draw my Punnett square, I have to think, how can I set this up so that my offspring will match the results that were described in the problem? And this is the only way you can set it up so that half of your offspring will have pink flowers and half will have white flowers. It's a cross between a pink flowered snapdragon and a white flowered snapdragon. Notice that anytime I have an R and an R prime in the same box, I always write the R first. So if it's heterozygous, like this, I would never write R prime R. You always put the R first and then the R prime. And that's just kind of one of those picky little details. If you do it the right way, you'll be less likely to get confused. So incomplete dominance, again, just means that a heterozygous individual has a phenotype that's in between the two homozygous phenotypes. In other words, pink is sort of halfway in between red and white, but it's not because red and white are blending to make a pink allele. There's no such thing as a pink allele. It's just that when both a red allele and a white allele are present, the phenotype is intermediate. To me, one of the most interesting things to talk about in the genetics unit is how blood type, what we call ABO blood groups, are inherited in humans. This is a great example of a concept called multiple alleles, because unlike most genes, the gene for blood type has not just two different alleles, but actually three different alleles: big IA, big IB, and recessive little I. It's also a great example of codominance, because if you have a person who inherits a dominant big IA and a dominant big IB, well, they can't both be dominant, right? They obviously, if you know, there's a reason why we're calling this codominance. It's almost like they're both trying to be dominant at the same time. So what happens in that case is they're both expressed equally, and they both contribute to the phenotype, which is called type AB blood, AB blood type. So like most things in genetics, this probably doesn't make a whole lot of sense to you until you start looking at some examples. But notice this dominant big IA allele codes for a type of carbohydrate, a sugar molecule called carbohydrate A, which is going to be attached to the surface proteins of your red blood cells. Big IB codes for a different carbohydrate, has a different structure. And recessive allele little I codes for neither of those two carbohydrates. And so there's three alleles, multiple alleles. That's what that means. But of course, you as an individual can only have up to two of the three, right? Because you have one mom and one dad, and so you can only have two genes for blood type. But in the gene pool, in the whole population, there are three different kinds of alleles that are out there that can be passed down. So here's how this works. The big IA allele, again, that's a dominant allele. It codes for a sugar that has a particular structure called carbohydrate A, and that sugar is attached to proteins on the surface of your red blood cells. This is a red blood cell, the types of cells that carry oxygen throughout the body. And so a person whose blood cells look like this has type A blood. Now some folks have type B blood, which means they have a different shape carbohydrate attached to the proteins on the surface of their red blood cells. Notice how these blood cells look different. So this person has type B blood. That's a different shaped carbohydrate. That's a different allele that codes for that. And now it is possible to inherit a big IA from one parent and a big IB from the other parent, which means that your blood cells have both carbohydrate A and carbohydrate B. So your blood cells look like this, and we would say you have AB blood type, which is somewhat rare, at least in the United States. AB is the most rare blood type. The most common blood type is type O, at least in the North American population. Now, it does vary a little bit as you go to different geographic locations. I think in Asia, type O is more rare and type AB is more common, but in our neck of the woods, type O is the most common blood type. Type O means that you are homozygous recessive, little I, and so your blood cells do not have either the A carbohydrate or the B carbohydrate attached to the cell surface proteins. And so this is called type O blood. A little interesting side note, the reason blood type is important, medically speaking, is because your immune system recognizes only the carbohydrate type that you have on the surface of your red blood cells. So for example, if I have type A blood, and this is the structure that my blood cells have, this is what my immune system recognizes. But if I am ever in an accident or I have surgery and I'm in a hospital and they give me a transfusion of type B blood from a donor or type AB blood from a donor, my immune system does not recognize carbohydrate B, the little circle. My immune system does not recognize that because my blood doesn't have that. So my immune system will react to this incoming blood as a foreign invader. It's a structure that is not recognized, that should not be in my body. So the only thing the immune system knows to do is to kill that cell. And this attack on the foreign blood proteins creates massive coagulation, blood clots forming throughout the body, organ failure, heart attack, stroke. It's a major, very often deadly medical mistake. However, if I have blood type AB, and specifically if I'm what's called AB positive, I don't want to get into the whole positive negative aspect right now because that's a whole nother rabbit hole, but just take my word for it. If I am AB positive, I am the universal recipient. I can receive a blood transfusion from any human on the face of the earth without any risk of rejection or coagulation or any problem. On the other side of the coin, if I'm O negative, and again, I don't want to get into what is meant by positive, negative right now, although that's easy to research if you're interested. But if I'm O negative, if that is my blood type, I am the universal donor. I can donate blood to any human on the face of the earth. Just a little interesting side note there. Okay, out of everything that you see on this little table, the most important thing that I want to call your attention to is this genotype row. Because this, after all, is a unit on genetics, right? So I want you to understand how you inherited your blood type from your parents. So if you have type A blood, there's two possibilities. Perhaps you inherited big IA from both your mom and your dad. In that case, you're homozygous, big IA, big IA. Or it's also possible that you inherited a big IA dominant allele from one parent and a recessive little I from the other parent. Either way, either one of these two genotypes will give you the phenotype of type A blood. Likewise, either of these two genotypes will give you the phenotype type B blood. Big IB, or big IB, little I. Either way, this is your blood type. Now, there's only one way to inherit type AB blood. You have to get a big IA from one parent and a big IB from the other. It's the only way a person could have type AB blood. And there's only one way to have type O blood, and that's little I, little I. Now remember, this one's pretty common. You might think, well, golly, that seems like that would be rare. But it turns out that there are a lot of recessive little i's in the gene pool. In other words, a lot of folks that are walking around in the population with type A blood have this genotype. And a lot of folks that are walking around with type B blood have this genotype. There's a lot of people walking around with little I, even if they don't have type O blood. So that makes it pretty likely that when a mom and dad get together and produce offspring, that those little I's are going to come together and make a homozygous recessive type O child. Let's look at an example problem. Oh, look at this crazy name. Agnes. That's an old-fashioned name. Agnes has type A blood and has just given birth to fraternal twins, Betty Sue and Jim Bob. Now, if you don't know, fraternal twins, of course, are born at the same time, like all twins, but they're not identical. One could be a boy, one could be a girl, as is the case here, and they're no more genetically alike than any other siblings. They just happen to be in the womb at the same time, born on the same day. All right, so Agnes has type A blood. She's just given birth to Betty Sue, who has type B blood, okay, and also Jim Bob, who has type O. So interesting right off the bat. Neither kid has the same blood type as mom. That's not that unusual. Show the cross between Agnes and her husband Joe. Well, we don't know squat about Joe. First, we have to determine what is Joe's blood type, and then we're going to try to show a Punnett square to show the cross between Joe and Agnes. So these blood type problems are kind of fun. They're kind of like brain teasers, kind of like solving a puzzle. You have to take the information that's given in the word problem and kind of tease it out one detail at a time. And usually you can figure out the right answer. So let's see what we can do with this. So let's see. First thing I know is that Agnes has type A blood. So here's Agnes. I put her genes on the side of my Punnett square. Now, I know that if she has type A blood, she's either got to be big IA, or what else could she be? Big IA, little I, right? Let's go back to our little chart. If a person has type A blood, you see, they must either be big IA, or big IA, little I. Those are the only two possibilities. So she has type A blood. So right off the bat, even if I don't read the rest of the sentence, I know she has to have at least one big IA. Maybe I can figure out this other gene later. Okay, now I don't know anything about Joe. Here's Mystery Man Joe. He's just her husband. We don't even know his blood type. But if you read on, it tells us that one of the kids, the boy, in this case, Jim Bob, has type O blood. That's very helpful. Because if a kid has type O blood, the only way that can happen is little I, which tells me something about Agnes, and also tells me something about Mystery Man Joe. Both of those parents, dad and mom, have to have little I, right? The only way a kid can have type O blood is if both mom and dad have a little I. Okay, what else do I know? Well, Betty Sue, that's the other twin, the girl. She has type B blood. Her big IB had to come from somewhere. Could it have come from Agnes? Here's Agnes. Does she have a big IB? Nope. So that big IB that Betty Sue definitely has had to come from, you guessed it, her dad, Joe. So Joe is no longer a mystery man. We now know that Joe's blood type, that was our first question here, his blood type is type B. Joe has type B blood. And now we are ready to fill in the rest of our Punnett square, and we can now answer this question. What is the probability of Agnes and Joe's next child? They've already had two kids, but when they have their next child, what is its probability of having type A blood? Well, that's 1 out of 4. The probability of type B, 1 out of 4. Type AB, 1 out of 4. And the probability of type O, 1 out of 4. So this is a very interesting couple, Agnes and Joe. This is a man and a woman who could theoretically have 4 different kids with 4 different blood types. Pretty unusual, but totally possible given the information that we're dealing with in this problem. So let's let you practice one on your own. I'm going to show you the problem. I want you to pause the video, work the Punnett square, and then you can check your answer. Here's the problem. Mike has type B blood. Mike's mother had type A blood. Mike marries Lily, who has type A blood. Both of Lily's parents had type AB. Show the cross between Mike and Lily. Now, you're given information about Mike and Lily's parents to perhaps help you solve the problem, but all you're going to show me in your Punnett square is Mike and Lily. Show the cross between Mike and Lily using a Punnett square, and then calculate what is the probability that Mike and Lily's next child will have type A blood or type B, and so forth. Pause the video and give it a shot. Okay, at this point, I hope you have taken the time to work this Punnett square. Let's see how you did. So Mike has type B blood, so he's either got to have big IB, or big IB little I. Now you notice I went ahead and gave him a little I. I didn't just make a guess. I know he has to have that little I because his mother had type A blood. Now let's think about that for a minute. Mike's mom, Mike has type B blood. His mom has type A. So she could theoretically have been big IA, right? Mike's mother could have been big IA, but if she were, then Mike would have to have a big IA, and he can't because he's got type B blood. There's no way he's got a big IA, right? A person with type B blood cannot have a big IA. So if Mike's mom has type A blood, but she didn't pass down to him a big IA, the only other thing she could have passed down to him is little I. Therefore, Mike has to have a little I. Did you follow that logic? You might want to back up the video and think about what I just said again, maybe watch it again. But there's no doubt Mike has a big IB, and he also must have a little I, which he inherited from his type A mother. Okay? Now, Mike marries Lily. Lily, we know, has type A blood, so I've got to give her a big IA, at least one. Now, how do I know, and I know it for a fact, how do I know that Lily does not have a little I? Because both of her parents were big IA, big IB. There's no way she could have inherited a big IB from her parents, because if she had a big IB, she couldn't have type A blood. So if both of her parents were big IA, big IB, and yet she has type A blood, she must have inherited the big IA from both of her parents. I know she doesn't have a big IB because then her blood type would not be this. I also know she does not have a little I because neither of her parents had a little I. So there's a lot to think about. But once you get to the point where you can take the words in the problem and set up the Punnett square, the rest is super easy because now I know you can fill in the boxes, and hopefully you did that part correctly. What is the probability that their next child will have type A blood? That's 2. out of 4, which is the same thing as 1/2. And the probability that their next child will have type AB blood is also 2 out of 4, which is 1/2. In other words, it's a 50-50 chance. Okay, we have a lot left to talk about in this part 4 lesson, but I'm already at 35 minutes, and so I'm going to stop here, and I will continue discussing non-Mendelian traits in part 5 next time. Until then, guys, have a great day.